Hello, hello. This is BIO461 week 4. And we're going to be talking a lot about muscle today. When I'm talking about muscle, I'm talking about calcium. Calcium makes up muscles. There's two different types of muscles. There's skeletal muscle and smooth muscle. Skeletal muscle is like the muscle we can control. That's connected to our bones. The smooth muscle is a muscle we can't really control. So, for example, skeletal muscle triggers a swallow that we take. But smooth muscle is the muscle that takes that swallow that we do and does the rest without our control. And so that's a little bit about smooth muscle. We're going to kind of zoom in to our bicep today a little bit and talk a lot about what makes up muscle fibers in skeletal muscle. And so to start off, we're going to use this whiteboard that I have. And we're going to act like this whiteboard is our bicep. And this is what it looks like. And we're going to zoom into our bicep and talk a lot about our muscle fibers and the names of what the muscle fibers are made up of. I'm going to say a lot of names. I don't know what all the names mean, but that's just what they are called. And so our muscle fibers kind of look like this. Draw it out a little bit. Okay, here we go. Muscle fibers kind of look like this. The vertical line we call the Z-disc. The horizontal lines we call the nebulin. And so inside of these things, there's something called actin or G-actin that binds to them that produces muscle contraction, and we'll kind of get into this a little bit. And so to start off, let's see here. I kind of have a paper that tells me what I need to go over today for this video. So there's something called tropomyosin, troponin, and actin, and myosin binding sites. Myosin binding sites kind of look like this. So myosin binding sites are all over our nebulin, and they control muscle relaxation and muscle contraction and things like that. And so tropomyosin is kind of like a stringy looking thing. It kind of looks like this a little bit, but it binds to the myosin binding sites like this for our muscles to relax. So this is normal when our muscles are relaxing. So this is kind of what our muscles look like when we're sleeping and things like that, at least most of our muscles. And then there's this thing called troponin. I'm going to draw it out. Kind of looks like this. There's three components to it. There's I, T, and C. The I component is most attracted to actin. And so actin is all of this as a whole. The T is attracted to tropomyosin, so the squiggly line. The C is mostly attracted to calcium. So the way this works is the T is wanting to attract itself to the tropomyosin and pull it away from the myosin binding sites. And so if there's no tropomyosin there, then our muscles are going to want to contract. When troponin pulls off tropomyosin, we call all three of this troponin. When it pulls off tropomyosin, the C component wants to bring in calcium because it loves calcium, brings in calcium to the myosin binding sites, causes a muscle contraction, and our muscles flex. And so this is an ongoing cycle when you go to the gym. That's how it goes. You go to the gym, you do a bunch of bench press, you flex your muscles, you take a break, tropomyosin enters or attaches to the myosin binding sites, your muscle relaxes. So this is inside mostly our skeletal muscle, almost all of our muscles though. And so that's a little bit of how they work. And so I want to talk about exactly zooming in a little bit to the actin and kind of how our muscles contract. Where is calcium going? In our muscles, we have something called T-tubules. They're like canals that bring in calcium into the muscle to cause contraction, which is normal. So we're going to draw this out. There's something inside of the cell that I want to talk about called the endoplasmic reticulum, or called the sarcoplasmic reticulum. We're going to call it the sarcoplasmic reticulum, or the SR for short. Let's see. Just drawing this out so that we can understand a little bit how this is going to work. Okay, here we go. All right, here we are. So this big canal is the T-tubule inside of our muscle zoomed up. So like this line here is our muscle. Inside of our muscles we have cells, or buildup of cells. The cell, inside of the cell, is the sarcoplasmic reticulum, or the SR. T-tubules are sent down, calcium is sent down the T-tubules. Once there gets enough calcium, there's a calcium voltage channel that opens up. There's tons of calcium voltage channels throughout here. We're just drawing one. It opens up, and we call that the DHP receptor. And then from the sarcoplasmic reticulum, there's an RYR receptor that connects to the DHP receptor it's like a bridge for calcium. It brings calcium in, sends it into the sarcoplasmic reticulum, and that is how our muscles are made. That is how calcium gets into our muscles and things like that. Now, outside of the sarcoplasmic reticulum is the inside of the cell. So ICF, intercellular fluid. These little dots that I drew we call the circopumps. It stands for this big long name. It's like sarcoplasmic, endoplasmic, reticulum, something, something. But it pumps calcium out of the sarcoplasmic reticulum into the cell, and then our muscles contract when that happens because they're like, oh no, that's not very normal. So they flex. And so this is what happens to our muscles when we are flexing and things like that. And that is how calcium moves from the T-tubule through the DHP receptor. RYR connects to the DHP receptor. It moves into the sarcoplasmic reticulum. Once there's enough calcium, it moves out through the circopump into the cell. Causes contraction of our muscles. So that's kind of how that works. That's me breaking it down just a little bit. And so, let's see. Can I erase this? See if I can get it. Okay, now I want to talk a little bit about, let me check my little paper. I want to talk a little bit about the two different types of smooth muscle. So, let's see here. See if I can see. Give me 1 second. Okay, yes. There are two main types of smooth muscle. There's visceral smooth muscle, where the cells are connected by gap junctions and contract together, like in the intestines. And there's a multi-unit, where each cell contracts independently, like in the eye muscles. The smooth muscles are basically the muscles that are in your blood vessels and in your stomach. The mechanisms of contraction with smooth muscle, we've already talked about mechanisms of contraction with skeletal muscle and how that works. The mechanism of contraction for smooth muscle is when calcium enters from the outside of the cell, not from the SR as much, but then the calcium binds to a protein called calmodulin. This protein activates myosin light chain kinase, which is something called MLCK. Then MLCK phosphorylates myosin leads, myosin heads. And I'm going to kind of draw this all out a little bit. Oh, I messed up already. I'm going to draw it. Okay. Oh my gosh. Sorry, excuse me. Okay. I think I drew that too big. Okay. Anyways, the MLCK phosphorylates the myosin heads, allowing them to bind to actin. Let's see. I'm going to draw this out so that we can see. And once the myosin heads bind to the actin, then contraction happens slowly. It lasts a long time with very little energy use. And then when calcium levels drop completely, myosin phosphatase removes the phosphate, which stops contraction. So smooth contractions are slower, they're more sustained and regulated differently than skeletal muscle. So to draw this out a little bit, let me see if I can draw this out really quick and kind of explain. So the two big types, we call them two different cycles. There's a phasic and a tonic cycle. The first cycle kind of looks like this. Okay. So from what we saw when I was drawing the Z discs and the actin and the nebulin and the myosin heads. So this is the actin, this is the Z disc, there's tons of them. This is our muscle. The myosin heads have ADP, which is energy, attached to them. This is the myosin tail, it has a phosphate attached to it. When it goes through this process, through the first step, calcium, ATP, and MLCK are added, which binds, which binds the ADP myosin head to the actin, also adding a phosphate. So there's two phosphates now. This starts contraction in the phasic cycle a little bit. The second step is similar in a way, but not really. So phosphate leaves and ATP also leaves, stops the contraction a little bit. There's less energy. Oh, sorry, can't really see that. There's less energy. The third step is sorry, I didn't draw it out. So this is what the cell fiber probably looks like at this point. Still binded, uncocked, still phosphate added to the tail. Second step, there's ATP added. ATP added. This is just for smooth muscle. Cell fibers start to look like this. They are no longer binded to the actin, still have a phosphate attached to their tail. And then for the final step, it can either go back to what it was normally. That means the MLCP needs to leave. And not ATP, but just P needs to leave as well. Now it would go back. If our muscles want to keep contracting, then it can go straight from this step all the way to this one and skip the first step without any problem. This is when like women are giving birth. They don't want to go back to this normal step. They want to keep pushing. And so their muscles are going to keep pushing and go, and make this cycle happen just between these three steps and ignore this first step because this first step is relaxation. So we call that the phasic cycle. Now there is also the tonic cycle of smooth muscle. And smooth muscle can alternate between tonic and phasic cycles. There's not muscles that are one or the other necessarily. they are similar in a lot of ways. So the muscle fiber is going to look like this. There's going to be a myosin head that I'm going to draw that's filled with ADP. The tail is going to have a phosphate connected to it. The first step is going to be the same. There's going to be calcium added, ATP added, which is energy, and then MLCP instead. MLCP and MLCK kind of cancel each other out. So it kind of looks the same in that way. We call this tonic. Tonic in this cycle is going to stand for tension. So this is very tensious muscles, like a sore throat or something, when these things are happening and just constantly happening. So when these things are added, myosin binding site connects to, or myosin head connects to actin. There's 2 phosphates added now. Phosphate, still ADP. And then the next step is a little more complicated. There are 2 phosphates that leave. So there's phosphate 2 that leaves. And then ADP leaves, the myosin head. Trying not to mix up my words while I'm writing. And then MLCP enters. MLCP? Oh, sorry. This is MLCK up here. Excuse me. MLCK up here. And then MLCP enters down here, cancels out the MLCK. And then it cocks the myosin tail. Now, for the final step, there is ATP added, which is energy. And this brings the tension in our smooth muscles. There's ATP added. And so now our muscle fibers are going to look like this. They're still going to be attached to the actin. They're still cocked. But the ATP can't decide whether it wants to stay in the myosin head because the myosin head likes ADP. ATP is going in and out. And this causes tension, which causes a latch state, which we call it, which takes a long time to get back to normal. That's probably the best way I could say it, just normal. So I'm going to call this latch state. So this is tonic. So there's tonic and phasic. Our muscles can alternate between these 2 cycles. So that's a lot about smooth muscle, the muscle that we can't control very much. That's kind of what's happening in it. Let's see here. I'm going to erase this up. And we are going to talk about sources of energy. There are 4 different sources of energy. Let me list them real quick. Do I have it? Okay. I need a different color. Here we go. There is systolic ATP. The systolic ATP, this type of energy only lasts for about 1 to 5 seconds, and we use it up pretty quickly. Second form is creatine phosphate. This doesn't last for very much longer. It lasts for 10 to 15 seconds. Number 3 is glycolysis. And I'm not going to go through all of glycolysis. It's taught in Introduction to Biology. It basically is glucose in our bodies converting into energy. This lasts for about a minute. And then finally we have aerobic respiration. Aerobic respiration lasts for hours. Okay. I don't know if you can see this well, hopefully. These are the different forms. So these 3, the first 3, are what we call anaerobic types of energy, anaerobic energy used. This is aerobic respiration. And so aerobic respiration energy in our muscles is used for standing for long periods of time. I wrote this in order 1 through 4 is because this is how our muscles order these different types of energy. When our muscles run out of systolic ATP, it goes to creatine phosphate, so on and so forth. And so each of these things are used for various things. And so aerobic respiration, like I said, is used for standing. Glycolysis is used for running really fast, like a 100-meter dash. Same thing with these two. They're used for very quick bursts of energy. And aerobic respiration and type of energy is used for a long period of time. That's why it lasts for hours. And so next, I want to talk a little bit about muscle spindles, Golgi tendon organs. And these things kind of go hand in hand a little bit. So let me see if I can erase this. I'm erasing this. Muscle spindles are simply put, muscle spindles are found inside of muscles and they detect stretch from our muscles. Muscles also have like built-in sensors to protect and regulate movement, and so this is what muscle spindles are and things like that. So muscle spindles are found inside muscles, they detect stretch. Golgi tendon organs, or GTOs, they are found in tendons. Tendons are in between muscles. And Golgi tendon organs detect tension from the tendon. And so like in our ACL and things like that. And so muscle spindles, they trigger the stretch reflex. I'm going to talk a little bit about stretch and withdrawal reflex. Muscle spindles, they do trigger stretch reflex. It's when a muscle is stretched, it contracts automatically to prevent overstretching. So like if we're trying to stretch our hamstring and we're bending over to go touch our toes, our lower back is going to want to bring us up because that hurts our hamstring a little bit. And so we call that the stretch reflex. And so our muscle spindles trigger our stretch reflex. Or another example that's probably more common is when you go to the doctor and the doctor checks for your knee jerk reflex. And so they hit your knee and you kick your leg forward because you're hitting a tendon and it's triggering that stretch reflex. Now, Golgi tendon organs, like I said, they trigger relaxation if the tension is too high in the tendon. They basically prevent damage from too much force. So they try our best to prevent tears and things like that. Tears in ACLs, PCLs, and things like that. Now, I talked a little bit about stretch reflex. I want to talk a little bit about withdrawal reflex. This one's more easy to understand. It's like if you touch something hot, like a stove. Sensory neurons activate in your finger that activate motor neurons that will make you pull away super quickly in your arm, in your finger. They basically inhibit opposing muscles, and so that withdrawal reflex is like touching a hot stove and pulling back really quick. When the stretch reflex is like a doctor checking your reflexes on your knee and then your leg kicking from it. And so that's pretty much all I wanted to talk about today. A quick summary of everything that we went over. The secromere is actin plus myosin equals the sliding filament model that I drew out a little bit. And then the action potential is when calcium releases from the sarcoplasmic reticulum, contracts muscles. ATP sources or energy sources, there's the stored ATP, there's the creatine phosphate, glycolysis, and aerobic respiration. Smooth muscle is slower. It uses calmodulin instead of troponin, like skeletal muscle uses troponin to pull off that tropomyosin for muscles to flex, like I drew on the whiteboard. And then finally, sensory reflexes. They protect muscles and maintain our posture overall. So that's what I wanted to talk about from what we learned this week. Thank you.